Hi, my name is Dan Blankenberg. I'll be talking about scaling up analysis inside of Galaxy using metagenomics as a perfect example for this. So first I'll give a brief introduction to what metagenomics is. Then I'll step through an example of actually doing it. Then I'll highlight some challenges, some solutions, and some workarounds that I encountered. So metagenomics is the study of genetic material recovered directly from environmental samples. So this does not require any sort of isolation or lab cultivation. And this is very important because not all organisms can be cultured in a lab. And so if you require any sort of cultivation or culturing, you're going to lose a lot of information from your samples. There's generally two different types of metagenomics. There's a 16S RNA targeted approach, and there's a whole genome shotgun approach. And these environmental samples can come from any sort of environment. So it can be different soil samples, it can be different locations within a human body, or any other organism, for example. This is a general outline of the two different types. So you have, on the left-hand side, you have your 16S RNA analysis, and then you have a whole genome shotgun sequencing approach on the right-hand side. What's important to note about the 16S RNA approach is that it is an amplicon-based approach. And so in this case, you actually are doing a targeted PCR in order to just get the 16S RNA gene. And then you can use this gene sequences to identify your organisms. The reason that 16S is used often, and sometimes 18S can be used in other instances, is that while this gene is highly conserved, there are some hypervariable regions that allow us to actually differentiate organisms within the sample. In particular, the study that I'm working with used regions from the V4 variable region. So now that we want to actually go about doing it inside a Galaxy, there's the general steps where you want to do your quality control and preparation. Then you want to classify your reads. Then you need to do some sort of normalization or rarefaction, calculate alpha diversities, beta diversities, then actually perform downstream analysis and visualization. I want to thank Sarah Craig. She actually did all of the sequencing, all of the sample preparation and so forth for all of the samples in this particular study that I've been analyzing. And this is actually, at this point, it's a little over 434 pairs of data collected from fecal samples and also mouth swabs for mother-child pairs. And she has done a tremendous amount of work to generate this data. So, of course, when you're starting with any next-generation sequencing data, you'll need to do some quality control. But before you can even do that, you have to figure out how you're going to get all of that data into Galaxy. Right now, the easiest way to do this is to use the FTP uploading abilities inside of Galaxy. So you can push the files from your computer after you've extracted them or after you've obtained them, and you can just push them over to Galaxy into an FTP server, and then you go ahead and you can import them into your history. And it's very important that you you click on this, you click on Choose FTP File, then you click on this button to select all of them. But you want to also make sure that if you have FastQ Sanger files, that you set them to FastQ Sanger if you don't want to go through the process of manually grooming your files and verifying your files. This is really important because if you don't do this, you're going to have a hard time actually changing the data type of 500 files in your history. You're going to have to click 500 things or come up with a couple of other workarounds, which I'll address later on. And then basically you just click Start, and that would add the data sets to your history. Now, you don't want to run a workflow 500 times individually, so there's something called Dataset Collections. And so once you have the files in your history, you would click on this checkbox here in order to make these checkboxes appear. You can click all, and that would select all of them. And then for all of the selected items, you want to build a list of dataset pairs in the case where you have paired N data, or just build a dataset list, for example. And then you can get into this wizard platform here where you can actually use parts of regular expressions in order to define your left-hand read and your right-hand read, your forward, reverse, et cetera. Then you would click auto pair, you give a name to your collection, and then it actually adds it to your history. And now you have a single item, a dataset collection that you can pass through a workflow as a single unit. With quality control and preparing now inside of Galaxy, you can do, oftentimes this is done with multiplexing. If you have not done the demultiplexing on the platform, you can do this inside of Galaxy as well. But once you have your demultiplexed reads, you want to make sure you remove all the primers, the barcodes, any low-quality bases, and then you may want to filter to remove entirely low-quality reads. These are all optional steps, of course, but I think trimming to remove low-quality reads is actually low-quality bases from reads is actually quite useful. Then in the case of the analysis that we're performing here, we're going to use Kraken, which is a Kamer counting type of classifier. And we're going to do that on actually merged or joined paired reads. So you have your forward and your reverse read, and they're long enough and over a short enough sequence that you can actually merge them together and basically create one single contig. And you can do this in Galaxy here with a workflow. Once these tools are available on usegalaxy.org, this workflow will also be available. So this is a typical workflow where we do FastQC reports along the way from our original data, then after trimming, after joining, and so forth. In this case, we're actually using two different types of joining. We're going to join where we actually merge the reads together, but at this merge step, it's actually going to kick out your unmatched left and unmatched right-hand reads. We don't want to necessarily lose these reads, so we'll use a second run of a different type of joiner that will actually let us merge our reads together by providing ends between them. So we basically have the left-hand read, we have a string of ends, and then we have the right-hand read. And this is perfectly valid to do in Kraken because of the way that Kraken works. It will ignore any Kamers that have ends in them. And so we're basically trying to have the most specific reads by using the merging join, but then we don't want to lose any reads that are still high quality, but we're not able to be merged together. One of the problems with FastQC, it's not really a problem, but one of the issues is that if you have 400 data sets, you're going to have 400 HTML reports that you're going to have to walk through and look at. And that's not very practical to do. And so there's a small workflow that we can actually build up inside of Galaxy to take our collection of reads, do some filtering on the text output, and then actually do a join across all of those data sets and end up with a summary report that actually, so we have one tabular file that has basically the test results for all of our samples. So across the top, it's wrapped around here, but this is one line, and these are our sample names. And then along the left-hand side, we would have our test. And in this case, this is the adapter content. And so all of these values here, we can see that we have pass. And so you would have additional tests all along the left-hand side as well. So when you want to classify reads, sometimes this is called as binning. It's basically your best attempt to identify where these reads belong, what groups, what OTUs, what species. In general, there's two different algorithms. There's a taxonomy-dependent algorithm where you do some sort of alignment to a set of known sequences and species. This is what Kraken does. And then there's a taxonomy-independent method where you can bin your reads based upon clustering and so forth. And this does not require a database. And so if you don't have a database that's extensive enough, you'll miss reads when you're using a database approach. So this is just a schematic of how Kraken actually works. So you have your query sequence. It assigns reads at different taxonomic, different k-mers at different taxonomic levels. It uses these counts, and then that basically defines how you can assign your reads. What's important when you're using Kraken is what sort of database you're using as the choice you're using. Kraken has facilities to automatically create a bunch of different databases, and so they're built off of the RefSeq references. What's important to note is that there's a standard database that Kraken will make. If you want to analyze your reads using this, the machine that you run it on is going to need about 61 gigabytes of memory in order to analyze that data. In this case, we're actually just using 16S genes, and so we can actually create custom databases that only contain 16S sequences, and those databases are actually going to be less than 1 gig in total. And so that'll actually run a lot faster and use a lot fewer resources. So this is just, in general, the Kraken workflow that you can go through. And so we have actually special tools that allow us to run individual reports from Kraken across our collection, and then we can join on the columns. And we end up with the same sort of layout as we did before. We're from the FastQC report, but in this case, we have our samples at the top. We have our groups called on the left first column, and then across the center, we have our actual abundance counts for each sample. When you're doing any sort of analysis like this across large numbers of different samples, you definitely want to do some sort of normalization procedure. What's very common is to do a rarefaction, and this is good to make sure that you've sequenced deep enough in your sequences, and you can also use it to rarefy your sequences to basically the lowest number of the lowest community number that you have. So what you want to see in these curves is you want to see a nice flat curve. So for example, if you would only have to sequence up to this depth for this green one, if you only sequenced up to here on this red one, you're going to have to assume that you did not grab all the organisms out of your analysis. So here's an actual rarefaction curve from the set of data that I was working with. One thing you'll notice is that it's actually, there's a couple of samples that did not sequence very well at this point, and so it's actually, if you were to rarefy at this level, you're going to lose a whole lot of sequences. So what you might want to do in this case is either resequence those samples or remove them from your analysis so that you don't lose a lot of this information. Now, looking at that curve, it's not too easy to do. So this rarefied tool will also report the slopes of the curve. So you can actually then just do some filtering manually to figure out what organisms, or sorry, what samples might not be robust enough. You want to calculate various measures of diversity. There's two different types of diversity. There's alpha diversity and there's beta diversity. Alpha diversity is basically the diversity within one sample. And it's an estimate of community richness, how many organisms are really there. And we have a vegan tool available inside of Galaxy that will allow you to calculate these values. With beta diversity, this is actually diversity between your samples. And so you can use this to compare how similar your samples are between them. One of the big issues with handling metagenomic data is that just having the raw counts or the raw abundances is not that useful. You often have a whole lot of additional metadata, such as where the sampling site was taken from, relation to other samples, and so forth. And this is not handled very well in a lot of tools, as far as each tool itself has its own way to define the metadata and how to combine it. There is a nice format called the biome format that actually does a good job of integrating the metadata directly in with the counts in a relatively sane fashion. And we have converters that will allow you to convert between tabular files and biome data. Okay, so here in this example, we have the input for left C, and we've actually gone ahead and we have our abundance counts, and then we have one individual set of metadata. And so we can build this metadata up inside of Galaxy, and then we can perform an analysis using left C. And so here we can see that in this particular example, we have an early and a late set of metadata, and we can see that we do have some organisms that are differentiating our samples. You can do this, generate additional plots using left C, and you can use the mode will actually do all of the differential features generically inside of Galaxy. This actually creates a tarball, and you can download this to your computer to view all of them. For visualization, one really nice visualization tool is Finch. And so you can actually view your biome data interactively at Finch. Let me talk a little bit about the challenges that we have. So loading about 1,000 data sets into Galaxy is doable in the UI. You have to have a lot of patience. When you click on things and wait, it's going to take a long time for your UI to generate. The problem is that when you get to 5,000 data sets, you really can no longer use the UI. With FTP, it's very easy to transfer the files over, but the problem is that you're no longer able to import them into your history. Building the collection in the UI for 1,000 data sets works very well, and building a collection for just a list of data sets is okay, but getting that wizard to work, it has a lot of issues. So like I said, when the issues with the FTP upload is that the tool launches a single job for each file. So if you upload 5,000 files and try to import it, it's going to launch 5,000 jobs. And so the workaround is that you can upload files in smaller batches. You can use an API script. And if you're going to use an API script, you might as well have that script also create the collection. 2.5. So upload tools, it should be allowed to import multiple files per job. The underlying command line tool actually already supports it. Before the UI was revamped, that's actually what it did. So we need to enhance it to not, the UI to not crash. And so another thing we should allow is importing actually an archive of files where we can actually provide a manifest or something else that will instruct Galaxy to automatically import that, create that collection. So once you actually can do your analysis, you have a lot of problems when you have a large history. So first off, you could not view the history. Your browser would crash when it tries to load all of that data. Additionally, if you tried to view your list of saved histories, it would take over 10 minutes to view your list of saved histories. There's something I refer to as the button of doom that I would accidentally click on when it was added. When you try to view the options, I would just by muscle memory, I would always click there, but that would also crash my browser. But these things have been relatively solved now. So if you can't view a large history, break it up into steps, use a history for like quality control, classifying, and then downstream analysis. You might want to hide your intermediate workflow steps. There's other additional potential solutions that are being worked on, particularly by Carl, as far as allowing us to view additional large data sets or large histories. And so there's also issues with viewing your saved history page, for example. And the problem here is that it was actually generating these counts using Python instead of just querying the database. And so that actually loads very fast now. The button of doom is actually no longer a button of doom, and it's actually a very useful feature. So you can have a multi-history view, and it won't crash your browser any longer. Another big issue is if you have one job inside of a collection fail, then that collection is useless, and then you'd have to either manually rerun the whole thing or use an API script to remove those errored data sets. But just recently, John Chilton went ahead and added some collection operations. So now we can remove failed data sets. But it'd be great if we could just rerun those failed jobs and slip them back into the collection. Okay, sure. So your element identifier now, so the issue is when you have large numbers of files, in your history, you have these tool on data sets, and those names don't work very well. And so now if you're writing new tools, instead of using dataset.name, use dataset.elementIdentifier. Additionally, I talked about fixing those files before. And then another issue is to try to add post-job actions for that. We have a whole lot of other additional options for doing metagenomics now. We have a mother tool set that has now been greatly updated. We're generating chime wrappers. You're going to hear about frogs just shortly. And I just want to acknowledge everyone here, tool developers, committers and contributors, along with the whole community and funding sources.
I always like to ask you the hard questions, Dan. With the biome annotation, is that a fairly manual process per file?
What do you mean?
So if you're going to add stuff like the metadata, where you got the sample from, what it was sequenced on, et cetera.
Right, so if you have a tabular file that lists your samples and then the metadata values along with it, you can just use the add or create biome tool, and that'll just actually fix that into this JSON format for you.
Oh, that's nice.
So that works fine. A lot of times generating those tabular files is the hardest part.